Welcome to the deep dive. We're going to be looking closely at some bacteria today, specifically Klebsiella, Citrobacter, Enterobacter, and Serratia. You sent us a chapter from Principles and Practice of Clinical Bacteriology, and we're ready to dive into it, break it all down.
It's a big chapter for sure. A lot of important info in there, especially for medical microbiologists.
So let's get right to it. Who are these bacteria? What makes them tick?
Well, think of them as a family, right? Like all part of that Enterobacteriaceae family.
Okay.
Which means they share some basic traits. They're rod-shaped, oxidase negative, and they're facultative anaerobes.
Oh, facultative anaerobes, meaning?
Meaning they can survive with or without oxygen. Pretty adaptable.
For sure. But like any family, they have their own, quirks, differences.
Definitely. Like Citrobacter frundii and Citrobacter diversus are only 50% related genetically. Think about that, like distant cousins who barely share any family traits.
Wow, that's wild. So even though they're grouped together, there's a lot of variation within these genera.
Yeah, exactly. And this diversity plays a role in how they cause infections too.
Speaking of infections, these bacteria are generally harmless,
Yeah.
right, living in our gut. But how do they turn into troublemakers?
They're what we call opportunistic pathogens.
Okay.
They can cause infections when the conditions are right, especially in people who are more vulnerable.
So like if someone's immune system is weakened, these bacteria see their chance.
Exactly.
Yeah.
And Klebsiella pneumonia is a good example of this. It can cause a whole range of infections, especially in hospitals.
Yeah. We're talking those tricky nosocomial infections.
Yeah.
That really make a hospital stay even worse.
Exactly. And what's interesting is that Klebsiella infections can show up almost anywhere in the body.
So it's a real challenge to diagnose them just based on symptoms.
Yeah, it can be. It can be, especially with a bacterium like Klebsiella pneumonia that can do so many different things.
Now, I remember reading about Klebsiella pneumonia subspecies rhinoscleromatis.
Yeah.
Causes a chronic infection called rhinoscleroma. Why is this one so hard to deal with?
Well, this Klebsiella pneumonia subspecies rhinoscleromatis has this ability to survive inside macrophages.
Inside.
Yeah, those are the immune cells that are supposed to destroy bacteria.
Wow. So it's like hiding from our defenses, like a Trojan horse.
Exactly. And that makes treatment tough because the bacteria are shielded inside our own cells.
It's a real puzzle for researchers.
It is.
So let's talk clinical setting. How do you actually know someone has an infection from one of these bacteria?
Well, it really depends on the specific bacterium and where the infection is. Like, Klebsiella pneumonia can cause pneumonia, but the symptoms, they might not be that different from pneumonia caused by other bacteria.
I see. So you can't just go by the symptoms. You need those lab tests to confirm.
You need those lab tests, and that's where laboratory diagnosis comes in.
Okay.
For most of these bacteria, we use standard culture techniques to grow them in the lab, and then we can identify them.
So you're talking about like taking a sample from the patient, growing it on a petri dish, and seeing what happens.
Right, exactly. And we look at things like colony morphology, how the colonies look, biochemical reactions, and which antibiotics work against them.
It's like piecing together a puzzle, right?
It really is, yeah. In the case of rhinoscleroma, caused by Klebsiella pneumoniae subspecies rhinoscleromatis, we look at the tissue under the microscope.
Okay.
We look for Mikulicz cells. They're these foamy macrophages that contain the bacteria.
That's fascinating. It's amazing how different techniques are needed for each bacterium.
Yeah, for sure. And once we've identified the bacteria, we need to understand its epidemiology. How it spreads, who's at risk.
Okay, let's talk about that. Where are these bacteria hanging out when they're not causing trouble?
Well, Klebsiella, Citrobacter, and Enterobacter, they're all considered part of our normal gut flora.
Okay.
They're usually there in low numbers, not causing any problems, but they can become a problem when they overgrow or move to other parts of the body.
It's like those quiet neighbors who suddenly throw a wild party and wreck the place.
That's a good way to put it. And some groups are more vulnerable to these bacteria, like premature babies, hospitalized patients.
Right. Anyone with a weakened immune system.
Yeah, exactly. Their defenses might not be as strong. And here's something interesting. The half-life for carrying aminoglycoside-resistant Klebsiella pneumonia in elderly patients can be 100 days.
100 days.
Yeah.
That's a long time for these bacteria to hang around.
Yeah, and some people even excrete the bacteria for over 200 days.
Wow, that's incredible.
It is. It makes it even harder to control their spread, especially in hospitals.
So what can we do to prevent outbreaks?
The key is to break that chain of transmission. And that starts with basic infection control measures like Hand washing.
Hand washing, the golden rule of healthcare.
It is. It's one of the most effective ways to prevent the spread of these bacteria.
So simple, yet so powerful. But what happens when these bacteria do cause an infection? How do we fight back?
Well, the treatment depends on the specific bacteria and its antibiotic susceptibility pattern. We need to know which antibiotics will actually work against that particular strain.
So it's not just a one-size-fits-all approach.
No, it needs to be targeted. And unfortunately, Klebsiella species, they're known for acquiring and maintaining antibiotic resistance plasmids.
Right, those pieces of DNA that can spread resistance between bacteria.
Yeah.
Antibiotic resistance is a real problem these days.
It is. It's a big challenge. And here's the concerning part. Resistance to new antibiotics often emerges very quickly.
Like, how quickly are we talking? What affects that?
It really varies. Depends on the antibiotic, the bacteria. But some things that play a role are how widely the antibiotic is used,
Okay.
ways bacteria can develop resistance, and how fast those resistance genes spread. It's a pretty complex interaction.
It's like we're always in a race against these bacteria.
You could say that.
Yeah.
And we need to understand their resistance mechanisms if we want to stay ahead, develop new antibiotics, and use the ones we have responsibly.
That's a good point. So let's move on to Citrobacter.
Okay.
What's their story?
Well, this genus, it got its name back in 1932, and right now there are 11 recognized species.
11 species. Another diverse group of bacteria, it seems.
They are. Citrobacter species share some things with the other Enterobacteriaceae we've been talking about, but they have their own unique features too.
Like what?
Well, they're usually rod-shaped. Most don't have a capsule, although some C. friendii strains do produce the bicapsular antigen. They're motile, they can move around. That's because of their peritricious flagella, meaning they have flagella all over their surface, and some even have class 1 fimbriae.
Fimbriae, those are like little grappling hooks.
Exactly. They help them stick to surfaces. Now, biochemically, all Citrobacter species are lactose fermenters, but some C. friendii isolates, they can be late lactose fermenters.
Late lactose fermenters. What does that even mean?
It means they take a bit longer to break down lactose.
Oh!
And this, along with their ability to produce hydrogen sulfide, has sometimes led to confusion with Salmonella species.
So they can be tricky to identify.
They can, but we have ways to tell them apart, like the indole test. C. friendii is indole positive, and Salmonella isn't.
So it's all about those tiny details.
It is. It's those details that help us solve the puzzle.
Now, I'm curious about their ability to cause disease. Are they as troublesome as Klebsiella?
Well, to be honest, we still don't know everything about how Citrobacter causes disease. We know fimbriae play a role in colonizing the intestines.
Right.
But the production of the vicapsule and the E. coli O157 lipopolysaccharide, it seems more about confusing us in the lab than actually causing harm.
But they're playing tricks on us.
You could say that.
Sneaky little things.
Or,
But they can still cause infections, right?
oh yeah, they can. There's evidence that some C. friendii strains can produce verotoxin 2.
Verotoxin 2.
Yeah, a powerful toxin often associated with E. coli outbreaks. And this toxin has been linked to gastroenteritis and even hemolytic uremic syndrome.
Which can be really serious.
It is. Can lead to kidney failure.
So they shouldn't be underestimated. What about other Citrobacter species? Are they a problem?
Another one to keep an eye on is C. koseri. It's been linked to neonatal meningitis.
Meningitis. That's an infection of the lining of the brain and spinal cord, right?
That's right. And in these cases, C. koseri expresses this unique outer membrane protein.
A unique protein. What's that for?
We don't know for sure yet, but it could be how it survives and replicates within macrophages.
Wait, survive inside macrophages? Didn't we just talk about that with Klebsiella?
We did. It seems C. koseri has a similar trick for dodging our immune system.
Incredible. These bacteria have evolved some amazing strategies to survive.
They really have. It's fascinating.
So where do we usually encounter these Citrobacter species?
Like a lot of the Enterobacteriaceae, they're found in the intestines of humans and animals, and they're all over the environment. In soil, water, even sewage.
They're everywhere.
Pretty much. And C. koseri in particular, it seems to easily colonize the neonatal intestine, even establishes itself in the mother's or nurse's intestinal flora.
That's a bit concerning, given its link to neonatal meningitis.
It is. It shows how important infection control measures are, especially in neonatal units. We need to prevent transmission from caregivers to babies.
Makes sense. Are they as frequent a cause of infections as, say, Klebsiella?
Not really. Compared to Klebsiella species or E. coli, Citrobacter species, especially the multi-drug resistant ones, they seem to be more easily lost from the gut.
So they're not as good at sticking around.
Exactly. They might be there for a shorter time, less likely to cause long-term problems.
Okay, that's good news. But they can still cause serious infections, right?
They can. C. cocceri, it's a significant neonatal pathogen, often causes meningitis. And sadly, even with treatment, half of infants who get C. cocceri meningitis end up with long-term complications.
That's really sad.
It is. It shows how important early diagnosis and treatment are to try and prevent those complications.
Absolutely.
Yeah.
So how do we identify these bacteria in the lab?
Well, Citrobacter species can be grown from infected sites, no problem. But, as we talked about earlier, telling them apart from Salmonella species and E. coli, that can be a bit tricky.
It's like a bacterial puzzle.
Yeah, it is. We have to look at how they look on different agar plates, their biochemical reactions, and their antibiotic susceptibility patterns. All those things help us make a definite identification.
Okay. And are they generally susceptible to the usual antibiotics?
It varies, but Citrobacter species, they tend to be susceptible to ciprofloxacin and imipenem. But there's a unique thing about this genus. They carry these inducible cephalosporinases.
Inducible cephalosporinases.
Yeah, they're also known as MPC beta-lactamases. They're enzymes that can break down some antibiotics.
Okay.
And they're encoded on the chromosome, meaning it's just part of their DNA.
It's like a built-in defense mechanism.
Yeah, it is. And what's remarkable is that these enzymes, they've been found in strains that were isolated before we even started using antibiotics.
Wow, so they've had this resistance for a long time.
It seems so. It's like they were prepared.
So how do we treat Citrobacter infections? And how do we prevent them?
Well, for treatment, it all depends on the antibiotic susceptibility of the isolate. But usually, fluoroquinolones, imipenem, or amikacin are good choices. Depends on the infection, of course. And for prevention, it all comes back to good infection control practices, especially hand washing.
Hand washing, always hand washing.
Always. It's really our best defense against these bacteria. Now let's move on to another genus, Enterobacter.
Okay, ready for another deep dive.
Enterobacter is a large genus, 15 recognized species. But the ones that cause most human infections are E. cloacae and E. aerogenes.
So those are the ones to watch out for.
They are, and they're very active biochemically. In fact, E. aerogenes is pretty similar to Klebsiella pneumonia biochemically. But unlike Klebsiella, Enterobacter species are motile.
So they can move around more easily, spread more quickly.
That's a possibility. And they have other features that set them apart. For example, unlike Serratia, which we'll talk about later, Enterobacter doesn't have lipase, between 80 esterase or DNase activity.
It's all about those subtle differences.
It is. It's what helps us tell these bacteria apart.
Now let's look at their morphology. What do they look like under the microscope?
Well, they're rod-shaped, motile, thanks to their peritricious flagella. And they have class 1 fimbriae, those grappling hooks we talked about.
Right.
They ferment glucose, producing acid and gas. They're methyl red negative, Voges-Proskauer positive.
Those are all tests used to identify them.
They are. They help us differentiate them from other bacteria that are similar.
Okay. What about their ideal growing conditions?
They actually grow best at 30 degrees Celsius, but most of the isolates we get from patients, they grow just fine at 37 degrees, human body temperature.
They like our warm bodies.
Yeah, they do. Now, E. cloacae has a wide range of O and H antigens. And about 80% of E. aerogenes isolates have a capsule, but it's usually thinner than the capsule of Klebsiella.
A thinner capsule. So maybe not as well protected.
Maybe not. And here's a fun fact. Some Enterobacter species produce a yellow pigment when it's colder.
A yellow pigment? That's kind of weird.
It is. It just shows you how diverse these bacteria can be.
Okay, so what makes Enterobacter species pathogenic? What makes them cause infections?
Well, like with Citrobacter, we don't know everything yet, but it seems like fimbriae, those grappling hooks, are important for sticking to mucosal surfaces.
So fimbriae are a big deal for a lot of these bacteria.
They are. They're how the bacteria attach to our tissues and establish themselves.
Where do we usually find these Enterobacter species?
Enterobacter species are everywhere. In the environment, especially in soil and water. E. cloacae and E. aerogenes, they're also in the intestinal flora of humans and animals, and in sewage.
So just like Citrobacter, they're all around us.
They are, and both E. cloacae and E. aerogens are considered important nosocomial pathogens. Hospital-acquired infections. They can cause outbreaks in healthcare settings.
That's never good.
No, it's not. And other Enterobacter species have been linked to specific infections. E. sakazaki and E. hormeche, for example, they've been associated with neonatal sepsis.
That's a serious bloodstream infection.
It is very dangerous for newborns. And E. taylori is a rare cause of nosocomial infections.
So these Enterobacter species can be pretty dangerous, especially for vulnerable patients.
Yeah.
What about outbreaks? How do they happen?
Outbreaks have been traced back to a few things. Contaminated enteral feeds, those liquid nutrition supplements,
Right.
and dextrose infusions. It seems they like to grow in solutions with high glucose concentrations. Hospital cross-infection is another way they spread. Contaminated urinals, transmission via hands, especially in neonatal units.
Again, highlights the importance of infection control.
Absolutely. And just like Klebsiella, Enterobacter species can survive on hands. Hand hygiene is so important.
It always comes back to that, doesn't it?
Yeah. It does. It really is a simple but effective way to prevent the spread. Okay, let's talk antibiotic susceptibility. Are these Enterobacter species susceptible to the antibiotics we commonly use?
Correct.
E. cloacae and E. aerogenes are generally resistant to certain cephalosporins and amoxicillin, but most isolates are still sensitive to cefotaxime, ceftazidimum, sapirum, imipenem, gentamicin, and ciprofloxacin.
So we still have some weapons to fight them.
We do. However, just like Citrobacter, Enterobacter species can produce those chromosomally encoded cephalosporinases, those AMPA-C enzymes.
The ones that break down cephalosporin.
Exactly. And they can make treatment more difficult.
So we need to be aware of this potential resistance.
We do. We need to be careful and adjust our treatment strategies accordingly.
I'm seeing a pattern here with these bacteria.
You are. Antibiotic resistance is a big problem, and these Enterobacteriaceae are no exception. They've found ways to survive even when we use antibiotics.
It's an ongoing battle.
It is. We need to stay ahead of the game by understanding their resistance, developing new antibiotics, and using the ones we have carefully.
Let's move on to another genus, Serratia.
Okay.
Tell me about Serratia.
Serratia. This genus is maybe most famous for Serratia marcescens. You know that story about the red discoloration of cornmeal porridge?
Bleeding polenta.
Yeah.
I can't imagine what people thought back then. Finding your breakfast bleeding.
I know. It caused quite a stir. And that red pigment, it's called prodigiosum, it's led to some interesting mistakes in the past, like being mistaken for blood in sputum samples, led to some wrong diagnoses.
Wow. So this bacteria has a pretty colorful history, literally.
It does. It's even been used as a biological tracer to show how bacteria can spread through the air.
Clever. So what makes Serratia different from the other Enterobacteriaceae we've talked about?
One thing is their enzymes. Serratia species are indole negative.
Okay.
They produce lecithinase and lipase, and they're more likely to be gelatinase and deaminase positive than other members of the family.
So they have their own set of enzymes. That's how we identify them.
Exactly. There are 10 species in the genus, but the ones that most often cause infections in humans are S. marcescens, S. liquefaciens, and S. rubidea.
Okay, so those are the usual suspects?
They are. S. marcescens is the most common one, and it's found pretty much everywhere. Water, soil, plants, even the guts of rodents.
Wow, they get around.
They do. They can live in lots of different environments.
Let's talk about what they look like under the microscope.
Okay. They're rod-shaped bacteria. Most of them are motile. They can move around because of their paratrichous flagella. And some of them have type 3 fimbriae. Remember those, the attachment structures?
Yes.
They can form mucoid colonies. But most species don't have a capsule.
So not as well protected as some of the others.
Right. But they have other ways to survive. They can actually survive in disinfectant solutions.
Really? That's not good, especially in a hospital.
It's not ideal. It means they can hang out on surfaces and contaminate things like medical equipment.
Okay, let's go back to that red pigment, prodigiosum. What's the deal with that?
Well, certain biogroups of S. marcescens produce it.
Okay.
And S. rubidea and S. plumothica too, but only at temperatures between 20 and 35 degrees Celsius.
So it depends on the temperature.
Exactly. And the colonies, they can be completely red or have red centers or red margins. They're pretty easy to spot.
It's like they're waving a red flag.
You could say that. And most Serratia species have a particular smell. Kind of fishy or like urine.
Not very pleasant.
No, not really, but it can help us identify them.
So what makes them pathogenic? Why do they cause infections?
Well, apart from those type 3 fimbria, we don't really know what specific factors make them cause disease.
So there's still a lot to learn.
There is, but we know they can survive in disinfectants.
Right.
They can grow at relatively low temperatures.
Okay.
And they can stick to plastics. All of those things can help them cause infections.
So they're resourceful.
They are. And they're not usually part of the normal flora in adults, but they can colonize premature babies.
So they can be a problem in neonatal intensive care units.
Exactly. And interestingly, the production of prodigiosin doesn't seem to be directly related to their ability to cause disease.
Really?
Yeah. Up to 80% of the isolates we get from patients don't produce it.
So that red pigment might be more for show than anything else.
It seems that way. Okay. Let's talk about their epidemiology. Where do they hang out? How do they spread?
Okay.
Well, Serratia species are found everywhere in the environment. In water, soil, plants. They're sometimes found in the human gut.
Okay.
But it seems their main reservoir is the inanimate environment, especially moist surfaces.
Moist surfaces. What kind of surfaces?
Things like disinfectant and cleansing solutions, contact lenses, hand lotions, urinary catheters,
Wow.
parenteral fluids, mechanical respirators, even fiber optic bronchoscopes.
So they can contaminate a lot of things.
They can. They're good at finding ways to survive.
How do they usually spread?
Mainly through hands, although Serratia species aren't found on hands as often as some other bacteria.
So hand washing is still important, but maybe they're not as sticky as some others?
Maybe not.
And what kinds of infections do they cause?
Serratia infections can be sporadic or epidemic.
Okay.
They're not a common cause of bacteremia in adults, but they can be really bad in premature babies.
Makes sense. Newborns are so vulnerable.
They are. And they can cause urinary tract infections, pneumonia, meningitis.
Meningitis.
It's rare, but it can happen. Endophthalmitis, bacteremia, wound infections. There's even something called red diaper syndrome that's caused by S. marcescens being excreted in infant stool.
Red diaper syndrome. That's a pretty descriptive name.
It is. It's a good example of how the bacteria's characteristics can show up in unexpected ways.
So how do we diagnose Serratia infections?
Serratia species are usually easy to grow from samples using standard lab techniques, but there are a few things to keep in mind. Some S. plumithicus strains, they don't grow well at 37 degrees Celsius.
Ah, so the temperature matters.
It does, and we also have to be careful about contamination. They can be found in a lot of places in the environment, including disinfectants and lab equipment.
Makes sense. What about antibiotics? What works against them?
Similar to Enterobacter, Serratia species are generally resistant to certain cephalosporins and amoxicillin, but most of them are susceptible to ceftazidime, imipenem, cefpyrome, and ciprofloxacin.
So we have options for treatment.
We do, but susceptibility to cefotaxime and gentamicin, that can vary. And like the other genera we've talked about, they can also express those chromosomally encoded cephalosporinases, the AMPAC enzymes.
It seems like those enzymes are a common theme in this family of bacteria.
They are. That's why it's important to know they exist and to do susceptibility testing to make sure we're using the right antibiotic.
Can they develop resistance to other antibiotics?
But unfortunately, yes. Serratia species, they're good at acquiring and maintaining resistance plasmids.
Those mobile genetic elements that can spread resistance?
Exactly. And that can lead to resistance to antibiotics that used to work. We've seen cases of plasmid-encoded resistance to imipenem emerging in S. marcescens.
Imipenem resistance. That's concerning.
It is. It shows that antibiotic resistance is a constant challenge, and we need to be on top of it.
So what's the takeaway when it comes to treating and preventing Serratia infections?
Treatment should always be based on the susceptibility of the specific isolate.
Okay.
But ciprofloxacin, imipenem, and ceftazidime are often good choices.
Yeah, so we do have options.
We do. And prevention, as always, is about good infection control practices. Being extra careful with how we store disinfectants, plastic blood containers, contact lenses, all those things can harbor these bacteria.
It's all about stopping the spread.
Exactly. By understanding how they live, how they spread, and how they can become resistant, we can develop good strategies to prevent and control these infections.
Well, we've covered a lot today. It's amazing how much diversity there is within this one family of bacteria.
Yeah, it is. And we've just scratched the surface. There's so much more to learn about these bacteria, how they interact with the environment, and how they affect human health.
It's time for another deep dive. But before we do, let's see how much you've learned about these Enterobacteriaceae. Are you ready for a pop quiz?
Let's do it.
All right, 5 questions to test your knowledge. Don't worry, we'll go over the answers together.
Okay, bring it on.
Question number 1. Which of the four genera we've discussed is most commonly associated with nosocomial infections? And what percentage of hospital-acquired septicemia cases do they represent? Think about which one is most likely to cause trouble in a hospital setting.
Good one. It's all about remembering which of these is the ultimate opportunist.
Right. Question number 2. Describe the unique characteristic of Klebsiella pneumonia subspecies, Rhinoscleromatis, that makes it so challenging to treat and understand its pathogenesis. This one is tricky. Think about where this subspecies can survive and replicate.
It's a pretty unique survival strategy.
Okay. Question number 3. What's the main way Klebsiella pneumonia is transmitted? Think about those basic infection control practices that can prevent the spread of infection.
It's all about breaking that chain of transmission.
Right. Question number 4. True or false? Klebsiella pneumonia subspecies pneumonia, also known as Friedlander's bacillus, is a frequent cause of pneumonia. Explain your answer. Think back to our discussion of how what we used to think doesn't always match up with the latest research.
This one might surprise you.
Okay. And finally, question number 5. What's a distinctive clinical syndrome associated with community-acquired Klebsiella pneumonia bacteremia, and where is it mainly seen? This one had a really interesting geographical connection.
Think about those cases that happen outside of a hospital. And remember that unusual geographical distribution.
Okay, take your time. We'll be back in a moment to discuss the answers.
Can't wait to see how you did.
Me too. All right, welcome back to the deep dive. Ready to go over those questions?
I hope you've been studying up on your Enterobacteriaceae.
Let's see how you did. Question number 1. Which of the four genera we discussed is most commonly associated with nosocomial infections? And what percentage of hospital-acquired septicemia cases do they represent?
This one was all about thinking about those hospital settings. Which one thrives when patients are vulnerable?
It's Klebsiella.
Klebsiella is the one. 9% of hospital-acquired septicemia. It's a big problem.
Wow, 9%. That's a lot.
Yeah, it shows why infection control in hospitals is so important. Hand washing, disinfecting surfaces, all that.
Absolutely. Okay, question number 2. Describe the unique characteristic of Klebsiella pneumonia subspecies Rhinoscleromatis that makes it so hard to treat and understand.
This one was about remembering that unusual survival strategy. Not many bacteria can pull this off.
It's the living inside macrophages thing, isn't it?
Exactly. They can survive inside macrophages, those immune cells that are supposed to kill bacteria.
So they're hiding from our immune system.
Right, and that makes treatment a real challenge.
Okay. On to question number 3. What is the primary mode of transmission for Klebsiella pneumonia? Think about those basic infection control practices.
Think simple. What's the easiest way to break the chain?
It's hand washing, right?
Hand washing. Simple but effective.
So important. Question number 4. True or false? Klebsiella pneumonia subspecies pneumonia, also known as Friedlander's bacillus, is a frequent cause of pneumonia.
This one played on those old assumptions. What we think we know isn't always true.
So it's false.
It's false. Friedlander's bacillus, less than 1% of bacteremic pneumonia cases.
Less than 1%. Wow, so not really a big player anymore.
Nope. It shows how our understanding of bacteria changes as we get more data.
All right, last one. Question number 5. What's a distinctive clinical syndrome associated with community-acquired Klebsiella pneumonia bacteremia, and where is it predominantly observed? Remember that geographical connection.
Think about those infections that happen outside the hospital, and remember there was a specific location.
It was Taiwan, right? Something about liver abscesses and meningitis.
You got it. It's a syndrome involving liver abscesses, meningitis, or endophthalmitis, and it's mostly seen in Taiwan.
Interesting. Why Taiwan?
We don't know for sure. Could be environmental factors, genetics, who knows? Still a bit of a mystery.
A mystery with a global twist.
Exactly.
Well, there you have it. 5 tough questions about the Enterobacteriaceae. Hopefully you learned a thing or two.
Sounds like our listeners are on their way to becoming experts.
But our journey isn't over yet. We still have one more genus to talk about. Proteus.
Get ready for swarming motility, pungent smells, and even a bit of mythology.
Can't wait. Join us in part 3 as we explore the fascinating world of Proteus. We're back for the final part of our deep dive into the Enterobacteriaceae, and we've saved a real interesting one for last. Proteus. Maybe you haven't heard of them specifically, but you definitely recognize them in the lab.
Oh yeah, they're famous for that. Spreading all over those agar plates. It's called swarming motility.
Swarming, that's right, I've seen it. So let's get into this chapter from Principles and Practice of Clinical Bacteriology and learn about Proteus. What are they all about?
Well, like the rest of the Enterobacteriaceae family, they're gram-negative rods, facultative anaerobes too. They can survive with or without oxygen.
Okay.
But what they're really known for is their speed. They move fast.
It's like the fastest in the bacterial world.
You could say that. And this speed leads to that swarming motility you mentioned. They create these rings of growth on agar plates. It's pretty cool to see.
It is, like they're racing outward.
Exactly. And this swarming happens because the Proteus cells change. They differentiate into what we call swarmer cells.
Okay.
And these swarmer cells, they're longer and have way more flagella than regular Proteus cells.
So like they're souped up, ready to spread.
That's a good way to put it.
Yeah.
And Proteus has another unique characteristic.
You mean the smell?
The smell, yeah.
I've heard it's not very pleasant.
It's not. Kind of fishy, or like urine. That's because they produce ammonia. They break down urea.
So their metabolism is linked to their aroma. Interesting. So what kind of problems do these speedy, smelly bacteria cause?
Well, Proteus species are opportunistic pathogens. They often cause UTIs, especially in patients with catheters or other urinary tract issues.
So they take advantage when things aren't working quite right.
Right. And that urease they produce, the enzyme that breaks down urea, it actually helps them cause infections.
Oh, so?
Well, when urease breaks down urea, it makes ammonia.
Yeah, okay.
And ammonia makes the urine more alkaline.
More basic.
More basic, yeah. And that kind of environment, it helps Proteus grow. And it can lead to struvite stones.
Struvite stones, those are made of magnesium ammonium phosphate, right?
That's right. And those stones, they can get big, block the urinary tract, cause a lot of problems.
So their metabolism is directly linked to their ability to cause disease. Fascinating. Which Proteus species is the main troublemaker?
That would be Proteus mirabilis. It causes about 90% of Proteus infections in humans.
90%. So
Yes.
that's the one we need to watch out for.
Yeah. Proteus vulgaris is the next most common. And then there are a few others that are less common.
Okay, so how do we identify Proteus mirabilis in the lab? Is it that swarming motility that gives it away?
That's definitely a good clue. They spread really fast on agar plates, and they grow easily on standard media.
Okay. What about their biochemical properties?
Well, they're known for their rapid urease activity, and we have tests to detect that.
So you can actually see that ammonia production happening.
We can. Helps us confirm it's Proteus. Now, for the important question, what antibiotics work against these Proteus species?
Yeah. What can we use to fight them?
Good news is that Proteus mirabilis is usually susceptible to a wide range of antibiotics. Penicillins, cephalosporins, aminoglycosides, fluoroquinolones, trimethoprim sulfamethoxazole. Lots of options.
So we're not short on choices.
Not for mirabilis, no.
Yeah.
But just like with those other Enterobacteriaceae, antibiotic resistance is something we have to worry about.
So Proteus isn't immune to that problem.
Nope. Proteus vulgaris and other species can be more resistant. They often have plasmids with resistance genes.
Those plasmids, always spreading resistance.
It's a constant challenge, and that's why susceptibility testing is so important. Make sure we're using the right antibiotic.
Makes sense. Well, that was a great overview of Proteus. Fast, smelly, and they can cause some serious problems in the urinary tract.
They are. It just goes to show that even the tiniest organisms can have a big impact on our health.
Absolutely. And with that, we've come to the end of our deep dive into the Enterobacteriaceae family. We've talked about Klebsiella, Citrobacter, Enterobacter, Serratia, and now Proteus.
We covered a lot of ground. It's an amazing family of bacteria.
It is. We've learned about what makes them unique, how they cause infections, how they spread, and how we treat them. It's a lot to take in.
Hopefully our listeners have a new appreciation for these bacteria now. They're often overlooked, but they're really important.
I think they do. And remember, the world of microbes is constantly changing. New discoveries are being made all the time, so keep learning, stay curious.
Absolutely. There's always something new to discover.
Well, that's it for this deep dive. Until next time, keep exploring and keep those lab coats clean.